Welcome back to our Psych 351 Biopsychology lectures. I'm going to continue on with our discussion of Chapter 8, the control of movement. In this video, I'm going to focus on mirror neurons. Now, one thing I just want to caution you, the mirror neuron literature I think is a little bit premature in the strength of the conclusions it's drawing. This seems like solid research, but there's still a little bit of controversy. There's still some question about functionality, especially when it pertains to disease like autism. So it's a fascinating system. Just be a little cautious of the literature, like I said. I'm going to give you sort of what we are pretty sure we know, and you can use that as a foundation for future discussion of mirror neurons. By the end of this lecture, I do want you to be able to explain what the mirror neuron system is, as well as its hypothesized functions. I also want you to be able to tell me some of the stimuli that activate mirror neurons. Now here I'm showing you a summary diagram from a fairly recent review about mirror neurons. This is in Nature Reviews. That's a very solid journal. Here I'm showing you human, but one thing I really want to emphasize in particular with mirror neurons, when we talk about what a neuron responds to, individual mirror neurons, we have to do microelectrode recordings. That means I need to stick a microelectrode into a specific neuron. Now here I'm showing a human brain. This isn't done in humans. If I ask for undergraduate volunteers to let me stick recording electrodes in your brain, well, I hope no one would volunteer for those types of studies. Okay, so we're going to have to correlate it with MRI. There's a lot of problems with MRI. You need to be very careful about how you interpret that data. We'll talk more about that. But I just want to emphasize that most of what we know about mirror neurons is really done in non-human primates, especially rhesus monkeys. The bottom line is the mirror neuron system, as the name implies, is a system of neurons that is activated when a monkey performs a specific task or sees another monkey or even a human performing the same task. Okay, so if I reach for something, my mirror neurons will fire. If I watch you reach for the same thing, my mirror neurons will also fire. So people simplify a little bit, but I think it's a good start. You can think of the mirror neurons as an imitation neural circuit. Okay? Now, you know that imitation is how we learn. So if you think about developmental psychology, working with language, one of the biggest ways kids learn to do things is by imitating. So this may be a very, very important system for understanding human development. And you'll see that in addition to just motor movements, gross motor movements like reaching, this system is responsive to movements associated with emotion. So if someone else smiles, my mirror neuron system will also fire. So it may have something to do with empathy. That's the stuff where we're starting to get a little premature, and I think people over speculate a little bit, but it's a very interesting idea, and the evidence is looking like that's not an unreasonable way to think about it. Now, mirror neurons are located in two brain regions. There's going to be an anterior, that means front, and a posterior, that means back. Don't worry about memorizing the specific names or anything like that. I want you to know that we've identified two populations, an anterior and a posterior population of mirror neurons. Now you can see that these talk to one another. Okay, so you can see that they send information back and forth. In addition to talking to one another, obviously, if I'm going to respond to another animal grasping or another person smiling, I need visual input. Here, STS is going to stand for superior temporal sulcus. Don't worry about that, it's just a landmark. This has visual input, and this is going to send visual input into the first of these mirror neuron systems. This mirror neuron system is then going to send that information on to the anterior population of mirror neurons. Now remember, in human, these connections have not been literally demonstrated. We haven't done track tracing. It's impossible to tell, but functionally these connections are here. So it may be direct, it may be indirect. This information somehow does get to this posterior population. Now, one thing I do want to point out too, if you look at these names, all of these names, these abbreviations, are just abbreviations of locations. So one thing you'll see, if I look neuroanatomically, cortex is cortex. So if I look under a microscope and I look at cortex from here, and I look at cortex from here, it looks exactly the same. What distinguishes these two pieces of cortex is functional. Okay, so here we're starting to get into a functional anatomy and not physical anatomy. Okay, this is important. So when we talk about the language circuit coming up in a little while, we'll talk about the posterior language area. We'll talk about Wernicke's area. These areas aren't distinct anatomically. I can't look under a microscope and tell you, oh yeah, that's Wernicke's area. It's based on the function, based on what they do. Same with primary somatosensory and primary motor cortex. They look the same. Okay, so we're looking at functional, not anatomical distinctions. I want you to remember from this whole slide, basically two populations, an anterior and a posterior population. I want you to remember that they get visual input. PMC here stands for premotor cortex. Okay, so if I'm going to engage in a behavior, a motor behavior that imitates somebody else, obviously I'm going to have to move, and I'm going to need feedback from those movements to see how I'm doing in terms of imitating. Okay, so that's what these mirror neurons in the ventral premotor cortex are likely doing. Okay, so we have visual input, we've got some motor input, and then these brain regions here are going to help me generate a movement that resembles what I see, and I'm going to get feedback as to how I'm doing. Now, when we talk about various movements, so here we're going to be talking about grasping. There's a part of the macaque brain called F5. Okay, F5 is related to grasping. And this area contains mirror neurons. The reason, you don't need to memorize F5, but I want you to know there's a specific part of the brain that when I record from neurons in there, so this is a recording from individual neurons, spikes are action potentials. When the rhesus macaque grasps for this item, neurons fire. Okay, so I get action potentials in F5. Now, what's interesting about these and what makes them mirror neurons is the animal doesn't have to engage in the behavior in order for me to see action potentials. If the experimenter grasps for that same thing, we get a response. Now you'll notice it's generally not as strong a response as you get as when the animal actually engages in the behavior itself. But nevertheless, there's a response. Even though the animal's not moving, it's just sitting there. Okay? So the reason I spent a minute telling you about F5 here is it's specific for grasping. There's another area, F4. This responds when there's head and neck movements, but not when there's grasping. F5 responds when there's grasping, but not when there's head and neck movements. Okay, so in the cortex, there's going to be different populations of mirror neurons, but they're fairly specific. They're specific for grasping, specific head and neck movements. Okay, so I just want you to understand that it is organized separately. Okay, but what's I think really cool, again, is the animal engages in the behavior, I get a strong response. The animal just sees the behavior, and I get a response. Okay. Now, here is data from humans. I'm not going to go into too much detail here, but there's several different situations set up where basically the subject is going to see movement of the the index finger here. I'll just circle it. Sorry. We'll see movement of the index finger, and we'll have to move. There's some control conditions. This is I'm not going to go into those in detail yet. When the individual engages, imitates the behavior, you can see they get a response that's fairly strong. Okay, and this is using fMRI, bold fMRI. Okay, so when they looked at this area right here, again, don't worry about the names. When they look at the area, this is Brodmann's area 44. When you look at activity in Brodmann's area 44, and the subject imitates the movement, they actually move their index finger. Here's our baseline response. Now, you just ask the person without observing any movement, anybody else doing it, to move their index finger. Those neurons do respond. Okay, so we do get a response. So if we're imitating, so I'm doing the motor task and I'm watching someone else demonstrate that motor task, I get a very strong response. If I'm doing just the motor task, they just say move your index finger, I get a strong response, but not as strong as when I'm imitating. Now here's what makes it a mirror neuron, of course. If I sit there and I don't move my hand, but I see somebody else move their index finger, now I get a response. Okay? Now, as I said before, this is fMRI, so I'm looking at millions of neurons. Okay? I'm not looking at individual recordings like I was able to in the rhesus monkey and the other studies. So when we look in BA44, we see that there's a control region where we don't see activity. It shows that when you move your index finger, signal doesn't increase everywhere in the brain. It's specific to this Brodmann area 44. Okay, so imitation, where I see somebody move and I move, gives me the strongest response in BA44. If I just move, I get a response, but it's not as strong. What makes it a mirror neuron, again, and what I think is fascinating, is if I just see someone else move, I get activation of that part of the brain. All right. Now, what's interesting is that the action, that mirror neuron system, also seems to respond to intent. Okay, so here's two different situations. Here, an individual is grasping a cup of coffee, presumably to drink it. Here, a person is grasping that cup of coffee, but it's empty. Here, they're cleaning up. Okay, so here, It's a similar motion, and I'm grasping in both cases. But here, the motivation, the intent is to drink. Here, the motivation is to clean. So when you look at mirror neurons, some mirror neurons will respond to this situation, but not this situation. So what that tells you is this motor neuron circuitry doesn't respond just to the movement. It also responds to the intent of the movement. Now there's probably other motor neurons, other brain regions, other mirror neurons that respond to this situation, but not this. Okay? So there's specificity. Now, when we're doing fMRI studies in humans, we can't look at that specificity in terms of individual neurons. In the macaque, we can. Here, we have to hope there's a differential responsiveness. So, although it's not individual neurons, when you look at groups of neurons, again, some group of neurons in this brain region will respond more strongly to this stimulus than to the cleaning stimulus. Even though it's the same grasping movement, the region responds more when the intent is to drink. Now, people have tried to tie this back to looking at autism. And I'm not going to go through this in detail, but basically what they've shown is that we have less activity in the mirror motor system when people with autism look at faces showing expressions. Okay, less activity. And the sort of more severe cases equals less activity. Okay, so the more severe the case, the less the activity in the mirror neuron system. This is very interesting. One conclusion is that people with autism don't respond to these facial cues as strongly as people who don't have autism. The data is consistent with that. I'm going to throw out another hypothesis for you, though. I want you to think about these things skeptically, critique them, see whether there's other explanations whenever you get any experimental result. Hopefully you learn to do that in research methods. Here, people with autism avoid looking at other people's eyes. So they've done studies. Excuse my terrible drawing. Here's a person. Okay, if I look at I use eye tracking software and I have someone with autism, you'll see if you look where their eye goes, it kind of goes everywhere but the face, and in particular it completely avoids the eyes. People without autism, where do we look? Usually you look to the eyes first. Okay, so if we were to look at someone without autism, we're going to see something like this. Somebody with autism, you see something like that. Okay? Now, if I'm not looking at the stimulus, is my brain going to respond to it? Of course not. So this less activity may not be due to a problem with the brain per se responding to these stimuli. It may be more that the patient with autism completely avoids the stimulus. Most fMRI studies I've seen looking at these types of things don't control for that. It's very hard to control for that, obviously. So this is an interesting result. The data is what it is. I don't question the data, but what I do wonder is what it means. There's many interpretations, but the two most obvious, one is people with autism have brains where the mirror neuron circuits don't respond strongly to facial expressions. The other is Patients with autism avoid the stimulus, therefore the brains don't respond as strongly. All right, so that's the basics of mirror neurons. So again, I want you to explain what the mirror neuron system is and its hypothesized functions, really with imitation and empathy. That's really it. In terms of stating the stimuli that activate motor neurons, remember, it's not just a movement, it's not just a grasp. Intent is also important. If you want to know more about mirror neurons, there's a reference for a good review in all the slides. You can also email me. I think it's a fascinating system and interesting to talk about. And this concludes my series of lectures on Chapter 8, Control of Movement. As always, if you have any feedback about the videos, like to see more or less of something, let me know, and I will be happy to try to incorporate that. Thank you very much.